I am Laxmi Narayana B, Assistant Professor, Department of Medical Laboratory Technology, Yenepoya School of Allied Health Science, Yenepoya, deemed to be university. Today we are going to discuss about general properties of virus. Before going to the property, we should know about some medical terminologies. So coming to the specific learning objective, at the end of the session, you will be able to define virus and host, distinguish between virus and bacteria, describe the morphology of virus, classify virus, explain stages involved in viral replication. So coming to the virology, what is meant by virology? Virology is the study of viruses, their structures, classification, replication and interaction with the host organism. Viruses are the smallest unicellular organism that are obligate intracellular. Coming to the host, it is the organism that provide the necessary environment, resources and cellular machineries for the virus to infect, replicate and produce progeny cells. Viruses can infect animals, plants, fungi and even bacteria. When it infects bacteria, it is known as bacteriophage. Now we are going to see how viruses differs from bacteria. They are obligate intracellular, they possess either DNA or RNA, they are filterable. You know, virus is smaller than bacteria, they can pass through the bacterial filters. They cannot be grown on artificial media. However, they can grow in experimental animals, embryonic egg or tissue culture. They multiply by a complex method but not by binary fission as we see in the bacteria. Viruses do not have proper cellular organization. They do not have cell wall, cell membrane or cellular organelles. They lack the enzymes necessary for the protein and nucleic acid synthesis. They are not susceptible to the antibacterial antibiotics. Next, coming to the morphology of virus. So under this we are going to discuss about the nucleic acid, capsid, nucleocapsid, symmetry and envelope. So we are going to see what is virion. Virion is nothing but it is the entire virus particle which comprises of nucleic acid, either DNA or RNA, which consists of a protein coat called as capsid, together with known as nucleocapsid. Some viruses also have a outer envelope. Coming to the nucleic acid, viruses have only one type of nucleic acid, either DNA or RNA. Nucleic acid may be single or double stranded, circular or linear, segmented or unsegmented. So we will see some of the examples for enveloped double stranded DNA viruses and non-enveloped double stranded DNA viruses. Examples for enveloped double stranded DNA viruses are herpes simplex virus, varicella zoster virus. For the non-enveloped double stranded DNA viruses, adenoviruses and human papilloma viruses. Next, coming to the capsid. Capsid is composed of a number of repeated protein subunits called as capsomeres. We will see what are the different functions of capsid. It protects the nucleic acid core from the external environment. In non-enveloped viruses, it initiates the first step of viral replication by attaching to the specific receptors on the host cell, thus facilitate the entry of virus. Now we will see about symmetry. Depending upon the arrangements of capsomeres surrounding the nucleic acid, we can classify three different types of symmetries: isohedral symmetry, helical symmetry and complex symmetry. So these are some of the examples here. Next, moving to the envelope. Certain viruses possess an envelope surrounding the nucleocapsid. Envelope is lipoprotein in nature. It has both lipid part as well as protein part. Lipid part is derived from the host cell membrane and protein part is virus coded, which is made up of many subunits called as peplomers. Peplomers projects as spikes on the surface of the envelope. Some viruses may have more than one kind of peplomers. Some examples are influenza virus, which has two different types of peplomers, hemagglutinin and neuraminidase. Next, coming to the size of the viruses. Viruses are extremely small, which can vary their size from 20 nanometer to 400 nanometer in size. One of the smallest virus is parvovirus, which is around 20 nanometer in size, and largest being pox virus, which is around 400 nanometer. Because of the small size, viruses can pass through bacterial filters and they cannot visualize under light microscope. Size of the viruses can be determined by various methods such as electron microscope, ultracentrifugation, etc. Next, coming to the shapes of the virus. Viruses exhibit a variety of shapes based on their structural symmetry and composition. These shapes are primarily determined by the arrangement of their protein coat, that is nothing but capsid. And in some cases, we can able to see envelope, which is made up of lipid. Most of the viruses are roughly spherical in shape. Some other have bullet shaped, filamentous shape, brick shape and rod shaped. Rabies is a virus which has bullet in shape, Ebola is having filamentous, pox virus is having brick shape and rotavirus is having rod shaped. Next, coming to the classification of virus. Based on the nature of the nucleic acid, we can classify virus into two categories, DNA virus and RNA virus. DNA viruses which contain deoxyribonucleic acid as their genetic material. Some examples are Epstein-Barr virus, herpes simplex virus, varicella zoster virus. Coming to the RNA virus, they contain ribonucleic acid as their genetic material. Examples HIV, poliovirus and hepatitis A virus. Based on the presence or absence of an envelope, we can classify virus into two categories, enveloped viruses and non-enveloped viruses. Enveloped virus, they possess a lipid envelope surrounding the capsid. Examples HSV, EBV and VZV. Non-enveloped viruses, they lack an outer lipid envelope. Examples hepatitis A virus, poliovirus and adenovirus. Coming to the replication. So, viral replication is a process by which virus reproduce and generate progeny within a host cell. Virus do not undergo binary fission but undergo a complex way of cell division. Replication of viruses pass through 5 sequential steps. So, first step is attachment, second step is penetration, third step is biosynthesis, fourth step is assembly and fifth step is release. So, in the first step, it is attachment. It is the first and foremost specific step of viral replication. It involves receptor interaction. Viruses bind to the specific receptors on the surface of the host cell. Examples, HIV binds to the CD4 receptor on the T cell. The second step is penetration. Virus enters the host cell via one of the following method. It can be through direct fusion, endocytosis or direct injection of genetic material. Third step, biosynthesis. In this step, the following viral components are synthesized: nucleic acid, capsid protein, enzymes required for replication and regulatory proteins. Step 4, assembly. Viral nucleic acid and proteins are packed together to form progeny viruses. Assembly may take place in the host cell nucleus or cytoplasm. Step 5, release. Release of daughter virions occurs either by cell lysis or by budding through host cell. Usually non-enveloped viruses, they release through cell lysis. Going to the summary, so we know what is viruses. They are the smallest unicellular organism that are obligate intracellular. Host, it is the organism that provides the necessary environment, resources and cellular machines for the virus to infect, replicate and produce progeny cells. They are obligate intracellular parasite. They need some host cell for the survival. And we discussed about the size and structure. The size of the virus can vary from 20 nanometer to 400 nanometer. And based on the shape also we can able to classify. Then based on the nucleic acid and envelope, we classify bacteria into DNA virus, RNA virus, enveloped virus and non-enveloped virus. Coming to the replication, virus replicate through a complex process which consists of 5 steps: attachment, penetration, biosynthesis, assembly and release. These are some of the reference. Thank you.